to come here on stage, even those who are in the room. So Fanny, Dora, Klaus, Ruth, if you want to join. So that, did you leave NCL, Klaus? Did you dismiss? So can we recover your budget? So, okay, so now it's a question and answer time. So you have all these people, you know, who are, let's say, and Scott. Well, I didn't see you in the bag, so Scott, because yes, take a class when, okay, and Ruth, okay. So these people are, let's say, performing, let's say, the everyday characterization of your nanoparticles. So if you have questions, please, this is the time, very practical questions. No question is stupid, so please feel free to ask questions. Yes, please, gentleman in front, please.
Hello, and my name is Kyung Gang, and from Seoul National University, Korea. And I am a nuclear medicine physician, and we are setting up about the in vivo imaging using radionuclide and the PET, MRI, and PET CT, and SPECT, and the radiolabel, all the nanoparticles. And so, do you have any partners and who is doing the in vivo, looking at the in vivo biodistribution or efficacy on the accumulations and using the in vivo imaging?
Yes, we do have, well, it's fine. We do, as part of the description that I gave you, we do in vivo in terms of biodistribution, and this is through clinical standards. So that's where we provide as part of our report, as a final report. Imaging is an aspect that is combined to one of the satellite lab. We can actually couple that aspect. It depends on what imaging technique. At the moment, as you saw, we work on injectable for cancer. We don't have included a multimodality, which is part of the theranostic space, which will be probably in UNCL 2.0 from this perspective. But we have the imaging technique that can be available, and through the discussion with the sponsor, we develop the best match in terms of using the data, because the regulators are based in their criteria on quality, safety, and efficacy. So proving the efficacy is the first thing you develop in your SME or in your industry, because it's where you find the cutting edge or the unique selling point from your side.
Yeah, generally,
Sorry, hang on, because it's recorded.
In vivo biodistribution is normally just the mice, and many people are looking at the disease model, and so we are also making a disease model by myself. And so in Korea, so we are already under construction of the platform of the testing of the radionuclide using the new radiopharmaceuticals. It will be end next year, and so they equip with the GLP and the GMP and facility for it. And so, I just wonder that we can join some of the partnerships with you and one of the testing for that, imaging testing that.
I think we have EU NCL, US NCL, and K NCL, the Korean NCL. We can assist you. Has anybody has any other comment?
So I would like to comment about what you described looks like being a satellite lab. That means you bring a specific expertise, you have a specific expertise which is not available to prevent duplication. And so far there is no restrictions for the satellite labs for being European, American, or Asia, coming from Asia. So I think we could really look at the possibilities for you to apply. Well, how to become a satellite? A very practical way. That means you just send a short description of the technology or the set of technologies you have developed and who could be useful, let's say, for the evaluation of nanomaterials for medical application. And then we have a check just to make sure that it's not available. If it's not available within the consortium, then you are invited at the next General Assembly. So the next General Assembly will be end of May this year, yes, in two weeks' time, in Dublin. So then we can discuss immediately after if you want to consider this possibility. Any further question? Don't be impressed. Yes, Camille.
Hi everyone, thanks very much for the explanation. I'm Camille Rush from Eyes and Science. So I subscribed to the EU NCL a couple of months ago. So Eisen is going to be one of the technologies providers, I'd say, but it's not really a question. It's more like a remark that I noticed on your presentation, guys. Actually, how important is the concentration to you? And by concentration, I mean number of particles per units of volume for a given sample. Because I noticed that there was, unless I'm mistaken, maybe just one graph, which was Fanny's graph, that was giving the particle count versus size, which was actually one value given by cryo-TEM. To me, the concentration is actually quite a crucial value, especially for R&D early stages. So I would have expected to see that a little bit more, because you're talking a lot about the general characterization, like size distribution, ZD potential, which is totally fine. But what about the concentration? Is it going to come a little bit later on, or what's the deal with it?
Fanny? Wait for the microphone, or you can speak from here.
Well, first, thank you for your remark. It's actually at the moment in the cascade we do not have included a technique for calculating the number of particles per volume. So we estimate that based, for example, of the intensity that we get from the light scattering, but of course that is not, we are more focusing on size distribution than on concentration. So we do not have a quantification of the number of particles per volume at the moment. That is something that we are.
But you guys are intended to include that kind of a value.
Yeah. Go.
Let me clarify. What Fanny said on the we don't have, it means validated and approved by the US standard. What we do have as a partners, we have complementary technologies where we do, like I personally have an NTA, and I do NTA with an NS500, which with EMPA and other partner, we have gone through a previous project where we did inter-laboratory comparison as round robin. So we have that, but it's not been validated.
So why hasn't it been validated yet?
So I can answer your question indirectly in that the main parameter that FDA is interested in is not concentration and particle number, absolute particle number, but drug per particle, because that's the API that you're dosing. So we would use your instrument, for example, to look at batch-to-batch consistencies. If we saw a difference between those, that would raise a red flag, and then we could dive further into it. But it's not per se particle number or concentration of particles is usually not the main parameter that we address.
All right. Okay. Cool.
Yeah.
Thanks.
Any further question?
Okay, hello. This will be a very technical question. I read several papers on NCL of American, and I saw that Malvern is standard procedure for size distribution. And however, I have problem like when we have micelles and we have CMC, and in different concentrations we have different composition of the solution, and Malvern does change the refractive index strongly in case of solvent. In that case, how do you recommend to work with that problem?
So just to be sure I understand correctly, you mean with different concentration you have problems with the refractive index of the material, and so that...
No, not the material, the solvent, because when we change the concentration, we change the chemical balance between micelles and non-micelle. So we have changed the solution refractive index probably, and Malvern needs to input it value every time. So I think it influences strongly the measurement.
Yeah, absolutely. Even though that is a problem when you go to volume and number distribution, usually we do not trust the way Malvern in transforming the intensity distribution into volume and number. So that is not the right technology we would use to get a number-based distribution. So in that case, you do not need to have this information for calculating the intensity side distribution. This is one of the problems that you may have, and is not the only one, because of course you need to estimate also, for example, the refractive index of your material. There are other parameters that you need to know if you want to use their software. And that's quite critical, because in that case you have to be sure of your numbers. So I would suggest to use other techniques in this case to get the number-based distribution.
Okay, I hope that maybe there is a solution, but it's not so simple. Yeah, I'm using NTA for a comparison, but
besides that, of course, the technique is the one that is approved also by FDA, and but if you get complex systems or polydispersed systems, it's sure not the way to go unless you want just a very fast pre-screening and something that is very cheap. But even if you want to check different batches, that you would not get enough resolution unless you are using that coupled with fractionation techniques. So I would anyway suggest not to go really further into how to use the batch mode DLS, but to focus on other techniques that can give better resolution.
Any further question from the audience? Or if you are, I don't know if how many of you are developing nanomaterials for medical applications? Okay, I could. So, and just for my information, what is, let's say, the status of developments? Let's say, is it, let's say, late preclinical? I mean, are you ready to consider, let's say, the regulatory application for the clinical trial? And use the microphone, please, because it is recorded. Everything is recorded.
Sure. Okay. For us, it's very early stage preclinical, so we're some way off, but it's very useful to hear from you guys what capabilities are available. It's extremely useful.
So, may I jump on your comment? I think it was expressed by Simon, and well, a little bit like our US cousins, you know, NCL, yes, was initially designed, let's say, to perform characterization, develop, and so on. But by interacting with the applicants after a little bit more than 1 year, we realized that it was more urgent, let's say, to initiate a dialogue with the applicants at quite an early stage. So, okay, guys, these are the critical parameters, so you have to pay attention. And so my suggestion is don't wait, let's say, for the very last minute to apply. That means you can contact Simon or anybody from the core expert team at any time as soon as you have, let's say, a concept. Then you can, well, of course, it's for free, so you can initiate this dialogue, and then you can prepare, let's say, a better application. Because later on, once you will apply, it will move much faster. So
yeah, and in fact, I'm already talking to Simon at Adriele.
Good.
We've had initial dialogue. I've introduced the kind of thing we're trying to do. So what these sessions are useful for is for me to learn a little bit more about the stages of characterization that you guys can do. I now have that in mind and would expect to follow up with further contact in the next few months.
Excellent. Thank you. Can you tell us more about your nanomaterial?
Thank you for the presentation, first of all. I'm from the University of Basel here. My name is Stefan Siegrist, and in the group of pharmaceutical technology at the moment, we have various nanoparticles that we are looking at. My colleagues are looking at mostly the liposomal formulations, and on the other hand, we are doing extensive research on nanotoxicology and correctly characterizing these nanoparticles, as well as in vivo and in vitro, and we are using the zebrafish embryo model at the moment for biodistribution and early stage in vivo toxicity. And for me, it was just very interesting to hear what you guys are doing at the NCL, because most of the parameters that you guys are checking, we're doing as well. And I was actually hoping to see maybe if there was some sort of use of techniques that we do not do. This is endotoxins, what at the moment we are not looking at because we're too far off from actually initiating something that would go into preclinics. But then again, I'm asking if you have DLS or Malvern or whatever, do you guys correlate that to only cryotem, or do you use SEM, TEM, SAX maybe? Is that something that that you guys have looked into?
Well, for SAX, considering that our idea is to implement the technologies that can be easily exportable then for the sponsor. So the answer is no, not because we do not have it, but just because it would be really difficult then for the client to use that for a routine check of their material later on. And of course, we are using for size distribution different techniques, microscopy, that the technique depends on basically on the characteristic of the material. So of course on the material, on the size distribution that we are expecting, etc. And we also have seen that file flow fractionation coupled to DLS or to light scattering works quite well in resolving this particle size distribution of polydispersed materials. So this is something that we have already in the cascade. We do have an analytical ultracentrifugation also that we are using that is very nice to get both particle size distribution and also some hints on drug loading and drug release in complex media. That may be another technique that is nice and useful. And of course, NTA or TRPS are also techniques that we have in-house, but that at the moment they were not qualified, and so they are not officially part of the cascade that may be used in cases we think that it's needed. And of course, in that case, the sponsor should be aware that they haven't passed through the qualification and interlaboratory comparison, and so they were not, let's say, also US approved, not yet, and so they are in-house, but we are using them if we think we need. But for sure, at least the microscopy to visualize the particle plus a technique that can resolve complex particle size distribution are used.
Your in vivo model is quite interesting, but the zebrafish, from the regulatory perspective, is predominantly used when you look at angiogenic effect and basically VEGF activity, and so you're looking at intervessel segmentation and the fragmentation you can have. So it's a model that we adopt that goes farther into the development, and it's also an early stage development. So it has to be fit with the sponsor in order to best fit, depending on the API, but usually you have your API clearly developed. You're looking at the nano component and you're trying to assess that.
Right. May I add a word about, let's say, the analytical methods? NCL, I think like the US NCL initiative, they are not dedicated, let's say, to perform, let's say, the world-class characterization, let's say, at the best performance. The objective is to facilitate and to speed up, let's say, the application process towards the clinical trials. Right. So basically, we align our assays to the requirements expressed by the regulators. And for each assay, we try to identify, let's say, the fastest, cheapest, more robust, most repeatable method, because our best dream would be that some of these methods could be translated, as Fanny mentioned, to the sponsor's place to be used as a quality control in-house. So this is why, in order to perform, in some cases, and especially for some early development, we need to have, let's say, a more performant assays. And this is why we have the satellite labs. So some of these assays can be performed by our platforms, but some others can be performed only by the satellites. So in that case, and it is another, let's say, service offered by EU NCL, if during the dialogue with the sponsor we realize that you would need, let's say, some heavy analytical method, SACS, for instance, then we can put you in contact, if it's not already the case, with the right people, because it would help you in your development. So this is, but just to make a difference, you know, between, let's say, top-class characterization and routine characterization for a regulatory purpose.
May I follow up with another question? If you're saying, well, one of the part of what we are doing as well is we are working together with the Swiss Center of Human Toxicology, and together with them we are in the NanoReg for the Horizon 2020 project. There we have encountered the difficulty as well of, should we actually look at the Biocorona in terms of characterization or not? Is that something that, I don't know, that the regulatory bodies really want? Because at the moment, I'm just speaking for myself and my colleagues at the lab, but we're not quite sure if including the Biocorona in something like a safety assay is, if we are really there yet or not.
Yeah, sure. If you want to pick about the Corona, you are the king with the Corona.
So as far as a regulatory question, the FDA and EMA are not going to require that unless for some reason your specific concept relies on that Corona for the mechanism of action. As you probably know, we're very active in this field as far as the Corona. And I'll preface it with others are great friends of mine, but we disagree very, very strongly. Kenneth Dawson is a great friend. What we found as far as the Corona is, if I sample your blood and your serum in the morning and you've had a rough day, your Corona, and we've shown this, we published on this, is going to be completely different than in the afternoon when you're relaxed. It depends on what you had for lunch, and it is so dynamic for one specific person that using that to do any type of diagnostic or personalized medicine, we just don't view it as feasible. If you pool the samples, you can begin to see trends, as has been published. But we have a patient donor pool where we sample the exact same patient over years, and we see huge variability. So as far as as I said, a feasible assay that then is able to determine how long a particle will last or where it's going to be distributed, I just don't see that based on our empirical data. And I wish Kenneth was here so that we could debate it, but that's our position.
Well, thank you very much. Very clear statement. Do you want somebody else to comment on Corona? No.
Thank you.
Are you satisfied with the answer?
Yes, I am.
Okay. Any further comment or remark? So anyway, may I ask you for some help? That means if you can spread the rumor about EU NCL, because we realized that people developing nanomaterials here and there in universities, in startups, are not always aware of this service, which is available, which is for free because it is easy funded. So if you can just tell them, and so that because the more applicants, the more we will gain expertise, and the more we can share this expertise with you in return. And So I hope you have enjoyed this session. I would like to thank all the speakers and all the members of EU NCL. This is a really fantastic initiative. And so if you, one day you would like to apply, either as a sponsor or as a satellite or as a provider, anyway, we would be very happy to talk to you. Thank you very much for your participation. Thank you.